Hi, everybody, and welcome to Neuroscientists Talk Shop, UTSA's neuroscience research podcast. Today is November 14th, 2024. We're talking to Claudio Punzo, who is Associate Professor at the Department of Ophthalmology and Visual Science at the University of Massachusetts Medical School. Claudio's lab studies the retina and diseases of the retina, at least usually ones that involve the photoreceptors themselves directly. Hi, Claudio.
Hello, everyone.
And also with us is Erika Tatiana Camacho. Tatiana is our own retina metabolism and disease specialist. Hi, Tatiana, how are you doing?
Hi, thank you for having me here.
And me, I'm Charlie Wilson. So, Claudio, I'm fascinated by the situation from a scientific discovery point of view. So there's two layers of cells, the retinal pigment epithelium and the photoreceptor cells in the retina. They're immediately adjacent to each other. They're interwoven with each other. They depend on each other metabolically. The pigment epithelial cells feed glucose to the photoreceptors, and they feed lactate back. And also the pigment epithelial cells are responsible for cleaning up as the photoreceptors shed their skin, their outer segments. And so they need each other, and then at some point, the pair of them starts to die in some place in the retina. This is age-related macular degeneration. And there are other diseases where this happens as well, but this is the one we're sort of thinking about today. And so our first impression is to figure out who's the murderer and who's the victim. Right? What went wrong in one of them that ended up killing both of them? Because once one kills the other one, it kills itself indirectly. So from a scientific discovery point of view, it's tricky. It's hard to find cause and effect.
Yeah, it's kind of a perfect crime, right? Where you're like, who is the killer and who is the victim? And I don't know if we have a full answer to that question, because it's almost kind of a sequential steps of events, right? Because the two depend on each other metabolically. Energy flows from the choroidal vasculature through the RPE to photoreceptors, right? And then a certain amount of energy flows back, as you said, lactate, right? That can be taken up, that is produced by the photoreceptors, can be taken up by the RPE and by other cells in the retina. And then the RPE, by phagocytosing and digesting, right, the outer segment, it gets a lot of fatty acids, right? And it's burning fat, basically. And then eventually that interaction starts to fall apart. And then who is killing who is a very good question. I think as far as we believe to understand is maybe initially there is a reduction of glucose to the RPE from the choroidal vasculature, at least in age-related macular degeneration. Maybe due to the buildup of lipids, and less glucose forces the RPE to change its metabolism, send less to photoreceptors. Maybe that causes the photoreceptors to synthesize less outer segments and change the lipid composition. That causes the RPE again to get less nutrition from the photoreceptors and maybe send even less down the road, and then maybe it's going to become a cascade of events. From a chronological point of view, it's really difficult to say who dies first, because we know that the number of photoreceptors is going down before the RPE is really disappearing. So it's slightly shrinking a little bit. It's not a lot, right? And then eventually the RPE disappears in that area. And then if that disappears, then the photoreceptors really go down. So chronologically, you would say if the RPE dies, the photoreceptors die. If the photoreceptors die, the RPE does not have to die.
But the RPE, if it's not getting lactate from the photoreceptors, it would probably be okay if it switched to glycolysis as its own energy source. Is that right?
We would assume that that would be the case, right? It would be okay functionally and would survive fine.
But it would be killing its partner.
But it would be killing its partner, and because it's killing its partner, it's maybe killing itself slowly, because it's not getting other stuff it needs from its partner. It's this weird interdependence, right? Where in the model we have created, where we change the fatty acid composition of these photoreceptor segments, the digestion doesn't work in the RP well. So it's almost like it's eating something that is somewhat toxic. It makes the lysosome not function properly in the RP. So maybe what it's eating as part of the photoreceptor, it needs a certain composition to be really happy and healthy. And if the photoreceptors is kind of disappearing and becoming unhealthy, the RP is less happy, even though it can survive, maybe on just glycolysis and using the glucose itself. Well, glycolysis and then oxidative phosphorylation conception, right? Using the glucose itself, it could survive very well. And it does in diseases like retinitis pigmentosa, right? When the photoreceptors die, the RP does fine until all the photoreceptors are gone, and then it starts to de-differentiate and move into the retina. So losing the photoreceptors does not necessarily affect RPE health in that sense. The retinitis pigmentosa is the disease where the RP sticks around for quite some time and does pretty well with losing 90% of photoreceptors.
So that argues in favor of it as the culprit. But it's to do that, it would have to change its metabolic style. And you can detect that when that happens. That's the kind of stuff that scientists can measure. How active is some enzyme? How much expression is there of some protein? And those are the indicators of metabolic style.
Yeah, and we do know that the metabolism is changing of the RP, right? So there are indicators that RP metabolism is changing. There are indicators that oxidative stress is increasing. So we know these indicators. We know, not 100% the chain of events in a disease like AMD, right? And then the question is really, what is causing the long-term progression and cascade? But when you are at the late stage, right, it's very easy to see how the cascade would lead to these detrimental events. But how is it really progressing over time slowly before it gets to that late stage? Conceptually, one would say, well, the photoreceptors can die and the RP is fine, so the RP is killing the photoreceptors, which in the absence of the RP, they do die. In AMD, not sure if the RP is killing the photoreceptors. To a certain extent, I think initially it is killing the photoreceptors. And it may be that the RP is forcing the metabolic change, and then the metabolic change forces additional changes in the RP.
So maybe it'd be good for us to actually talk about what are the metabolic changes that are known to be happening in this situation.
So, I don't know how many of these metabolic changes have been really established, right, and are accepted as real facts, or how much is still speculation. The recent study that came out by a group at the NEI, Dr. Swaroop, which looked at methylation changes of retinal DNA, clearly attributes a lot of metabolic changes in the retina, right? As you have AMD and as you progress through the stages.
So can I try to clarify that a little bit? Because DNA methylation tells us something about epigenetic regulation of gene expression. It doesn't really immediately tell me what the metabolic change would be. I guess there's a chain of reasoning that says increased methylation is going to mean repression of some expression of some protein, and then that protein is going to go down in its activity. Is that how that?
Yeah, and they looked basically at which genes had these methylation changes, right? And a lot of these genes were part of regulation of metabolism.
So So regardless of whether methylation goes up or down, you can still say, oh, this is the family of things
that is changing. And so metabolism is changing as a consequence, right? And it's sometimes very difficult to understand if you have methylation changes, right, around the promoter, a gene. Well, is every change having a positive or negative, like is the expression going up or down, right? With which change exactly? It's not that you know that all the time, but you definitely know that these are genes that, that something is changing. So it's part of a certain pathway. It could be inflammation, right? It could be other things, but it seems to be around metabolism is definitely one of the hallmarks. So there are metabolic changes in the retina of these AMD patients, and these are adaptive changes at the end of the day, right? It's an adaptation to a disease state. Now, have we proven that these metabolic changes are in a certain direction and that direction is causative in our case with the work that we are doing? No, we have not proven that. The only way to really prove it at the end is, we succeed in a clinical trial and we say whatever we developed as a therapy fits the concept and the idea, and that's why it's working in a human case. But we think that we have a lot of convincing data that says, hey, maybe it is metabolism and adaptive changes in metabolism in the photoreceptors itself.
So my understanding is what we do when we're in that kind of situation is we take an animal model, we make metabolic changes in that animal, and then we see if we can reproduce the symptoms of the disease or some pathology from the disease. And is that a good way to go? Are we making progress at understanding it that direction?
That is a perfectly good way to go, yes. And you're correct that that's the standard way of going. We conceptually did that, right? We created an animal model that we believe reflects the disease. We have the disease pathology, and then we solved, or we identified, a target drug in that animal model. And if we treat it with our drug, it works. But because the human disease has no clear genetic etiology, then the question remains, have we really created the correct animal model?
So your animal model is a genetic modification.
My animal model is a genetic modification. The human disease is not a genetic modification, right? So have we created with our genetics the correct animal model? We obviously have changed the genetic modification such that we are shifting a lot of cell metabolism, right?
Yeah, so I was thinking the choice of the genetic modification must have been rational.
Yes.
Some kind of hypothesis.
Yes, that is correct. Yeah, the hypothesis was taken from analyzing human tissue sections, right, where we see in diseased patients metabolic changes, and we were like, oh, okay, we assume that this is what is happening. Let's mimic that in a healthy mouse and see what happens. Now we are getting disease, and it looks like it's mimicking for all intents and purposes, what we can assess, it's mimicking the human disease. Have we really identified the correct thing? I hope so, and I wish so. I'm not 100% sure, but I'm pretty confident.
We never are.
We never are.
The changes that you made, was it in RPE?
The changes that we made were in the photoreceptors. So we predicted that the adaptive response in aged individuals in photoreceptors to changes that occur earlier in the RPE is what really precipitates the disease. So we only, activated this later step, this adaptation of the retina, of the photoreceptors, and that was sufficient to precipitate everything. So there are clearly events that have to occur earlier, right, for this adaptive response to occur. And we know that these events are happening in humans. Do we know the exact sequence of these events? No. We have rough ideas. But we mimicked basically this later response in an animal model, and that was sufficient to cause everything. So that's why we think that that's a crucial event in the disease progression that happens at one point in a patient that has early stage AMD.
What's the nature of the response? I know we don't necessarily want to go into the complexities of metabolic pathways, but I think we could go some distance into that, because that seems to be the crux of it. Don't you think?
Fair enough. So I think that what they are trying to do is get a little bit more efficient in the uptake of glucose, right? So that whatever is coming through, they are more efficient at taking it up. And they are shifting the composition or the profile of the fatty acids, right? Let's say if you had 2% of this fatty acid, 5% of this, and so on, right? You're still synthesizing roughly the same, but you're shifting the composition. Maybe now it's 10% of A and only 5% of B and 30% of C. So they're changing the profile, right? As an adaptive response. And, that maybe comes with good things. It helps them to survive better, but it may come also with bad, unintended consequences.
What are some good things that might...
Well, I do think that the good aspect of it is that the photoreceptors may be more nimble in their energy use, right? And survive longer with less energy. The bad thing is that they are producing maybe more unhealthy crap that makes the RP sick. It's
I see. So in a low resource circumstance, the cell switches its metabolism to something that's more efficient, but has products that are undesirable.
Yes, it's almost like, if you don't have enough food, we have a drought or whatever, right? And we are like, okay, we are using these high caloric products that we know can feed the population, but they are not that healthy because we don't have enough veggies and whatever with it, right? But we keep people alive, but we make them more sick. So it's a little bit like that, right? It's like it's an adaptive response to stay longer alive, but because you're maybe not synthesizing other products, you're making the RP less happy and more sick.
So do we know for sure any of the bad things that are coming from this change?
No, we don't know. So that's still something that we have to investigate and figure out.
So how about the molecules that are controlling this decision? Because this sounds like, your analogy was to a society making rational decisions about feeding the populace. And so sometimes we look at a cell and we think, the cell is making rational decisions about things, about what to produce and what to reject. And that's, cells do seem to be like that. So what kind of mechanisms could actually be the decision maker?
Yeah, that's interesting. Cells are rational. If you put all the cells together and you create a human, you get a lot of non-rational.
That's true.
That's when you have too much rational, you end up with something irrational. So, a rational decision maker in the cell is this kinase mTOR, which regulates all different aspects of cellular metabolism, and it's a very fascinating gatekeeper of cell metabolism, of which we understand a lot and very little, because it does adapt metabolism due to a whole host of environmental stress, right? Obviously, amino acid availability and glucose availability being one of the number one causes, but then also oxygen availability, fatty acid availability, and it produces different responses depending on the different cell type the stress happens, right? If you have a fat cell, if you're starving, you're not eating enough food, right? Eventually your fat cells will degrade fat. That will be in part regulated by the kinase mTOR. Right? your neurons is not going to degrade itself because there is no other outcome for it.
Mhm.
It's death or survive, right? So your neuron may upregulate protein synthesis while your muscle cells at the same time will downregulate the protein synthesis and will start to break down the proteins in your muscle to convert it into glucose, to put it in your bloodstream, right? So that you have enough glucose so that your neurons can survive. So the same physiological stress, like starving, will activate and downregulate mTOR activity and cause different responses in different cell types because they have to adapt differently. So it's a very interesting gatekeeper where in cell culture you can try to figure out what happens under certain condition, but only in the human body or in the context of an animal can you really understand how different tissues and cells react to that common stress, right? So I think mTOR is one of the central rational gatekeeper. We don't always understand rationally what happens in which cell type under which condition.
It's fascinating that this one complex of molecules could produce completely different results in different cell types.
Yeah.
There must be an elaborate network of contextual molecules that tell it what needs to be done.
Yes, there are a lot of different molecules that sense a lot of these stressors, right? And then feed into that kinase as an input. And the kinase then activates or represses a lot of other molecules to basically come out with that central response, right? So it is just one of the big gatekeeper where a lot of information comes in and a lot of information goes out. But some of the responses go directly from oxidative stress, right, to trying to adapt to it, where it involves mTOR, but it is also mTOR independent, some of the aspect, right? The final outcome has often mTOR dependent and independent input. Because it's such a complex response, as you say. So there are a lot of other players that, directly read out also these inputs and then cause a certain response in combination with the kinase mTOR. So it's not, one central hub, everything goes in and everything goes out. There are direct pathways, but that then require also the input of mTOR, right, to come up with the complex adaptive response of the tissue under certain stress conditions.
So the changes in gene expression are changing that network that mTOR is embedded in. Is that right? Of molecules that are transporters, for example, or molecules that perform, that burn energy or that create energy.
Yes, yeah. So in age-related macular degeneration, that's what we believe is happening, right? That there are changes in gene expression in molecules that process glucose, right? That synthesize fatty acids, that tell you how much fatty acid, which composition of fatty acid, that those things are changing as a response to the adaptation, right? And we really believe that we can pass out downstream, right, which are the good aspects and which are the bad aspects. And if we can keep the good and repress the bad, we may be able to significantly delay the disease, right? And that's kind of the goal of the research, to say, okay, the adaptation has to happen because the tissue and the cells have decided that that's the best way forward. But with, the good aspects come these bad aspects. And if we can pass it apart and say, we'll repress the bad aspect and just let the good happen, maybe we have a chance to actually really delay the disease and help people that have the disease.
So for people who don't do this kind of work, like me, how do you do it? I mean, how do you alter one of these chemical pathways at all?
So what we are doing, once we have identified the chemical pathway, what we are doing is then try to determine, well, what is the target gene, and then see, well, is it a small molecule that we would want to inject, right, like a drug, an inhibitor, Is it a virus approach where we permanently change the gene expression by some trick? Or are we using nowadays this new technology which uses siRNA, which is small interfering RNAs, right, for which the Nobel Prize was just given recently, this year, a couple of months ago or weeks ago. So, we basically decide, depending on the target that we have to hit, well, what is the best approach? And what we really take into consideration when we are trying to do that is also what works therapeutically as a translation, right? So, it's one thing to do an experiment in mouse and say, oh, this works. But if you never can translate it as a therapy in human because it's impractical, then maybe we are not going down that road if we have another alternative, if we can establish the proof of concept with a better drug, right? So if I have a small molecule that is an inhibitor, but I have to inject it every day, well, I can do the experiment in a mouse, but I can't make it a human drug.
Inject into the eye when you
I can't go to a human and inject the drug every day. The only thing I can give a human is a pill every day, right? Now, depending on the gene, if that pill has too many side effects, because you're trying to target only the eye, but the pill gets everywhere, right? If there are too many side effects, well, it doesn't work as a therapy. So if the gene and metabolism is something that is in common to your entire body, right? So everything you take that changes metabolism is going to have a lot of side effects. So we really want to deliver it to the eye. And then if it's injected, well, then the virus would be good, or the RNA therapeutics would be good, because we know from experience now that that stuff is going to last about a year after an injection. So it's a very translatable approach in terms of human therapy, right? So we have to think what works in a clinical setting. The patients can come easily once a year and have a checkup, right, and get also the drug. That works fine. They have to come every month. Kind of works, that's you're hitting the limit there every month. The benefits have to really outweigh the negative side effects, especially when you're talking about a population that is older, 75, 80. They can't come every month to the clinic. Maybe they don't have somebody that drives them there, right? And if the benefits of reducing disease progression, if they don't feel it very much, right, if it's not significant, they may just think, well, I'm not going to go to the doctor. I'll skip it. It's not worthwhile. Once a year works easy.
Yeah.
So we think about all these aspects when we are trying to develop a therapy as to, what is the best approach depending on the gene that we need to target, but also what works translational in terms of putting it in a patient rather than just a mouse.
So a small molecule approach is mostly interacting with some enzyme or some protein, right, and changing its function, whereas the genetic approaches actually change the level of the protein being synthesized.
Generally, the virus or the siRNA changes the levels of the protein, and the biggest problem with the small molecules is that most of them don't last long. Right? They get washed out very fast from the system because they don't interact with the protein even if they physically interact and bind, right? It's a permanent binding, because there is often a turnover of the protein. There is also a turnover of your drug, right? And it just disappears. And the effect is, in most cases, short-lived. There are not many small molecule inhibitors that last, for more than 2 weeks. The effects last for more than 2 weeks. So that's one of the big problems.
I'm always amazed that there are so many small molecules that target enzymes in metabolic pathways. It's almost any one of those enzymes has something that blocks it. Where did those all come from? Those were created by people like you, or they're made by pharmaceutical companies, or?
Well, most of them obviously, most of them come from nature, right? And they were identified by pharmaceutical companies. And they come from a whole variety of natural producers can be sometimes, fungi that try to protect themselves from bacteria, plants, and other living things that produce these small molecules that as a defense mechanism, right? So, and pharmaceutical companies have screened, I don't know how many different plants and fungi and all the small molecules they produce, right? And basically have run large assays to identify these inhibitors, right, of enzymes, of kinases.
And the logic is just if there's an enzyme that's involved in metabolism, sooner or later we're going to
there's going to be something that inhibits it, because somebody in nature will think, no, you benefit from that enzyme, and because I'm competing with you, I'm going to create an inhibitor. Yeah. I mean, it is true, right? You're going to find something in nature that is going to somehow inhibit that enzyme.
One of the most remarkable ones is rapamycin itself, which was in some kind of fungus on some island somewhere.
Yeah, the Rapa Nui Islands, I think, Rapa Nui, right? Or the Rapa Nui or the people. I don't remember.
But amazing for it to be found.
Yeah.
And this is an mTOR inhibitor.
Yes.
And so important in these kinds of experiments, I suppose. Is it used a lot?
So it is used a lot. Rapamycin is used. It's in clinical use. It's a clinically approved drug for immunosuppressant organ transplantations. Right? I don't know now if they have how many different other drugs there are that they use for immunosuppressant, especially in organ transplantations. It's a drug that is very well tolerated.
Isn't that remarkable?
It is remarkable.
Because mTOR seems like it's the rational actor that's deciding.
It is. But, it's fascinating. The drug kind of disassembles the complex, but it's very difficult to get 100% disassembly, right? So maybe you are left over with 10% activity, right, rather than 100. So... And that's why we think it's very well tolerated in humans, right? And different cells require different baseline levels. So it does conceptually work very well. If you were to knock out the kinase mTOR in all your cells, this would come in a few weeks. But then if you use rapamycin, the system tolerates it because you can't, I guess it's almost impossible to take so much drug that you would have 100% of the complex disassembled, right? And you have nothing functional there.
And actually, many people think that it's some kind of longevity drug, and there's an underground of people taking it on their own to increase their lifespan and so on.
Yeah, I don't disagree. There is that aspect, and I think it does work. It is a miracle drug. I don't know if I would go the route of rapamycin if you really want to have longevity. You can also go the route of, fasting 1 day a week, or just having a little bit caloric restriction works equally well. And they have shown that, the changes, the mTOR changes with caloric restriction and rapamycin are slightly different. So, the outcome is longevity in both cases. But I think, as far as I remember, the benefits with caloric restriction are better than with mTOR inhibition. So, if you're talking about long term. But it is a fantastic drug. It does things that we don't understand, right?
So I definitely don't understand it, because it sounded in one case, in one cell, it was going to do something completely different from what it did in another cell. I mean, mTOR does something different in the different cells. And so blocking it would seem to be doing dramatically different things in different cells.
So
how could there be a universal effect of it?
So one of the good aspects about blocking mTOR, right, once in a while, it's kind of like if you go for a run, right? You go for a run, you cause a lot of oxidative damage to your body, right? Free radicals. But the benefit of that is your body has to start to turn over the garbage, right? It's like you cleaning up your room and making it tidy again. And you're at home and have a little bit mess, and the mess is getting worse every day. You're like, yeah, I'll clean it up at one point, and here comes Tor, puts the hand on the table, and it's like, now it's time to clean up, right? And if you take rapamycin, you're kind of forcing the cleanup system as if you're going to take a run, right? And you're kind of forcing that cleanup in your body. And cleaning up the cell is a good thing, right? Gives you longevity because you're getting rid of all that oxidative damage that has occurred but was maybe not cleaned up efficiently because there was also too much energy flowing in the cell because we tend to overeat, and have too many calories. So the garbage is slowly coming in, but it's not going out, and it's just piling up. And rapamycin is doing that turnover. In a neuron, it's a little bit different, right? Because neurons tend to be metabolically more nimble. They don't store that much energy. They take it from the blood glucose, and that's why we have constant blood glucose levels, right? So they need regeneration as well, but other cells in your body need it even more than the neurons. And we keep mental acuity for quite a long time, even though physically we are regressing much faster, right? We can be mentally very active at the age 70 or 80 or 90. Maybe we can't run around anymore. So, it's a little bit different for the rest of your body than for your neuronal system. And that's why the reaction is also different, right? But obviously, our neuronal system in some cases suffers of neurodegeneration, Alzheimer's and other things. But then the whole system is affected. But that's most likely because of a different cause.
Well, thanks. We strayed a little bit off of the retina, but that was really interesting, I thought. And I really appreciate, Claudio, having you here. Thank you very much.
It was my pleasure coming, and thank you very much for having this chat with me.
Thanks, Tatiana.
Thank you. I didn't ask any questions. Just a quick question. The system adapts, but there's a adaptation only lasts for a certain amount of time before it actually all collapses.
Yes.
In your work, I know you tested for a long time. Do you check if there's a critical point where, like even doing this small interference RNA, well, it lasts for a year, but if you do it again, how often can you do it before it's just not going to work anymore?
So, so we think that, the RNA interference that we are using to reverse some of these changes should work, and you could re-inject it, administer for the rest of your life, right? At a certain interval. The adaptive changes that then lead to the collapse, right? The collapse being geographic atrophy or neovascular pathology, that one is very random and stochastic, and we do not understand it. Besides, it's tracking with the density of the photoreceptors. We really don't understand it. But it's, when it happens somewhere, it's not that we can reverse that, but we hope that we can prevent more of these occurrences to happen, and we hope that we can slow down the progression once it happened, right? Slow down the progression or the spread of it, the growth of these lesions. So does that answer the question?
Well, kind of, but also back to Charlie's question, because the effects are different in different cells, right?
Mm-hmm.
And so, well, it might be doing good in some cells. It might not be doing so good in other cells. So you might actually be, as you're trying to slow down the progression or trying to reverse it, like reverse the effect on photoreceptors, say, in other cells, might it be actually doing harm in other neurons?
That is possible, right? And it's a very good question. We know that the knockout mouse model doesn't have a lot of phenotypes, but that doesn't say that, if you're doing it in a human as a therapy, that you wouldn't have any effect on any other cell type, right? And the reason being is there's also a big difference between having a genetic knockout where you never had a gene, but you have a secondary gene that functions almost the same, right? Because then you can have, from birth and development, more expression of that. As when you have two things there and both are needed, and then suddenly you remove one, all of a sudden, right, the whole system starts to freak out because it was used to having it. And obviously the idea is to just reduce the levels. And we do not know yet what the effects will be in other cells. We suppose from all we know that it shouldn't be too detrimental. But it's a question you never know. Even when you do safety studies in a monkey, at the end, right, you don't know how it reacts in a human.
This has been Neuroscientists Talk Shop.